Okay, in this video lecture, we're going to start chapter 7. It's titled Bacteria and Viruses, and so we are kind of getting into animals. So you can see that we are breaking down this chapter into 3 sections. What are bacteria? And we'll discuss bacteria and archaea. Section 2 looks at the good and bad of bacteria and how they interact in nature. And then section 3, we talk about viruses. So I'm actually going to break up section 1 into multiple lectures here, actually 2 lectures, one covering bacteria and the other covering archaea. So the characteristics of bacteria is that they are microscopic. Okay, they're small. You need a microscope to see them. They're also prokaryotes. And remember, prokaryotes means like before the nucleus, they don't have a nucleus. Okay, so these are organisms that were around before the nucleus developed in the tree of life. They are one-celled, unicellular, no nucleus or organelles, and they live in almost every habitat, even on your skin. You are covered in bacteria. There's bacteria inside of you. The surfaces that are surrounding you right now are covered in bacteria. So here I have a hair follicle, and you can see bacteria stained with red. This is cilia in your nasal passages, and dyed blue is the mucus. You can see that the mucus is trapping some pathogens here. So when you sneeze, it's the cilia's effort to try to expel or get rid of the bacteria. Structure of bacteria: they have a cytoplasm, jelly-like substance inside the cell. DNA, it's coiled and circular. It is not in a nucleus, no nucleus here. A cell membrane, which is green, surrounded by a cell wall, which is in yellow here, and then a capsule on the outside, which is usually a kind of a mucus covering that prevents or assists the bacteria in avoiding, making them more resistant to antibiotics, making them stick better to their surfaces. They're small, 1 to 5 micrometers, and 3 shapes that you can find bacteria in: round or sphere shaped, rod shaped, or spiral shaped. Because bacteria are found everywhere and almost in every environment, they have quite a few different ways that they get food and energy. So there are some bacteria that are decomposers. They feed on dead organisms and waste products. There are some bacteria that are parasitic, which means they live in the host and they steal nutrients. There are some bacteria that are photosynthetic. They make their own food from sunlight. And if an organism, or in this case bacteria, does not require oxygen, then we call it anaerobic bacteria. So you usually find these guys in the stomachs of large mammals. Aerobic bacteria means oxygen is needed for them to survive. Movement: flagella, a long whip-like tail used in movement. That could have one flagella, could have multiple flagella. Your book also talks about pelis and how they just twist and move in their environment as ways to get through their environment. Reproduction: they copy themselves through process of binary fission. So the end result is 2 genetically identical cells. Now I throw conjugation in here. Conjugation is not a form of reproduction, but it does increase genetic diversity. So how it happens is you get 2 bacteria, they form a bridge between each other, and they kind of exchange genetic material. And the end result is you end up with a bacteria cell that might have more genes or different genes, and as a result you increase genetic diversity. Why is this important? Well, when bacteria cells just do fission, okay, through asexual reproduction, they're just making clones of themselves. So there's really no genetic diversity because the offspring are just like the parents. But if you throw conjugation into the mix, now you have some new genes that could be expressed. This is actually how bacteria are becoming antibiotic. Thank you. They're becoming resistant to antibiotics through conjugation. So this is a diagram showing binary fission. Copy the DNA goes on opposite ends of the cell, and it pinches in 2, and you end up with 2 identical cells. So this is an E. coli bacteria going through binary fission. This is showing conjugation. So 2 different bacteria cells here. They form a bridge, okay? Conjugation bridge, I think is what your book calls it, and they exchange genetic information back and forth. Now, Bacteria cells also have the ability to lay dormant for maybe months or centuries. And how they do that is they form an endospore around their chromosomes to protect it. So think of a jawbreaker with multiple layers. So an endospore is basically many, many layers going around their genetic information to protect itself from intense heat or cold or drought. And then when conditions are ideal, then they will shed these coats and then become active again. So that does it for 7.1 bacteria notes. Your assignment is a prokaryotic coloring sheet. All the answers can be found on that assignment, and then you have to make sure you color the prokaryotic cell. In that last paragraph, it tells you how to color it.